Now we have a cargo vesicle with no coat. Before we fuse it to the Golgi, so since this is COP 2, I want to mention in passing how the trafficking vesicle gets to the Golgi. And it's definitely not a random diffusion process. There is in fact a network of microtubules that link those two organelles, and in fact links pretty much everything going from the nucleus out into the periphery of the cell as part of the cytoskeleton. And it's usually the case that the vesicles travel along those microtubules, and that process of traveling is motorized by motor proteins, dyneins and kinesins, that use ATP hydrolysis to energize the actual translocation of the vesicles. I don't think I'm going to have time to talk about those motor proteins specifically, but I did want to mention them and to point out that in some cases the RABs are actually associated with that microtubule interaction and the motor proteins, so they're part of that process as well. But anyway, let's assume that that process has happened and it has brought the vesicle that we've just uncoated in the previous slide to near the surface of the Golgi. At that point, what happens here is going to happen. This diagram is also not specific to COP2, but again, we could make it so relatively simply, by just telling everyone that this is RAB1. We just decided, and that this is that 1P that I mentioned on the previous slide. So those steps now make this a specific fusion event. And the way fusion works generically pretty much describes all fusion events, regardless of the coat. So at this point we can be generic and be completely accurate about COP2. So the steps really begin here with an inactive form of RAB, which is again GDP bound, stabilized through the interaction of a protein called GDI in the cytoplasm. So this is the state that RAB would be in during the coat formation and the translocation of the coated form of the COP2 vesicle. Once you remove the coat, then that leads to the dissociation of RAB GDP from GDI, its conversion to the GTP bound form, and its association with the membrane of the trafficking vesicle. And that association is transient but significant enough, strong enough that it will last until this vesicle gets to the surface of the Golgi, as it turns out. So the snare has been uncovered as a result of the uncoating, and here's the cargo receptor. So the way RABs work is by specific pairing with proteins on the target membrane, which in this case will be the Golgi, but if you wanted to keep it generic, it could be wherever this vesicle is supposed to fuse. So this protein, the RAB effector, is the flip side to the specificity determined by RAB. That is, RAB1 will have its own specific RAB effector, and RAB6 would have a different one. And as a result of that distinction, RAB1 containing vesicles will not fuse to the ER. They will only fuse where they're supposed to, the Golgi, as a result of the fact that that's where the RAB effector is. So that specific pairing is the first step in the licensing or the approval of the fusion steps that happen next. It's referred to by your text and many people as tethering. It's the first step in the overall fusion reaction. This can be a very, very long protein. They're not all, it's a family of different types of conformations, but the RAB effectors can be 10 times the diameter of the vesicle, so they stick way out into the cytoplasm, basically grabbing the RAB. And if that's the appropriate interaction, then the steps proceed to the docking step, which involves bringing the vesicle much, much closer to the surface of the target And if this is a COP2 vesicle, this again would be the Golgi. And if that's true, then there'll be a specific family of T snares on the membrane, and they will react specifically and very, very strongly with the V snare of those COP2 vesicles. So you end up with a structure that looks like this. This is a coil-coil interaction involving multiple very, very long alpha helices. And that interaction is almost as strong as a covalent bond. It's about as tight as any non-covalent interaction you will see in biology. So, and it's specific in the sense that this has to be the appropriate V snare and this has to be the appropriate T snare. Otherwise, the process will not continue. So even if this is the appropriate match, if this isn't, then fusion will not proceed. But let's just say that these are the appropriate snares to form that coil-coil. Then now you end up with the very close approach of this membrane and this membrane, and that leads to fusion. So in terms of the overall goals, we have now deposited the cargo, if it's a soluble protein, into the lumen of the Golgi. If it's a membrane protein, then it would be embedded in the Golgi membrane. And just to sort of wrap up the procedure, we can't stop here because we want to have this process continue as soon as the next cargo vesicle comes in. So there's a little bit more to it than that diagram indicates. And what is involved in reversing the interaction of the snares is shown on this diagram. I like this one because it actually indicates that water is here. It's one of the few diagrams where water is not just assumed. But here's that interaction between V and T snares, just again a generic diagram in this case, that leads to this space becoming much less as the water gets excluded, so that there's a series of intermediates in which one of the leaflets of each membrane system fuses first, and that leads to an intermediate that looks something like this. As those snare interactions tighten, you end up with an extended region of the luminal membranes, and that leads to their fusion, so that you end up now with the continuous lumen between the vesicle up here and the Golgi down here. And these snares wrapped up in very, very tight twist ties. And that's shown in a more pleasing diagram, I would say, over here. So there's the fusion event. This is that complex that is so tight that you've got to use ATP to get it separated. So there's a protein called NSF that catalyzes that reaction using ATP, associating with this knot of snares, and as the ATP gets hydrolyzed, the two types of snares, V snare and T snare, get separated as a result of that catalytic mechanism, and then you can start the process over again.